All right, chapter 14, metabolic diversity of microorganisms. In section 1 here, foundational principles of metabolic diversity, we're going to be touching on the different chemical slash enzymatic slash metabolic reactions that will be covered throughout the rest of this chapter. Now we have to keep in mind that laws of thermodynamics apply. Okay. First law, energy cannot be created or stored, just transformed from one form to another. And second law, during that transformation, that changing of one to another, you're going to lose some. Now, you do the change all at once, take it from A, move it over to B, big amounts of energy. All at once, you're going to lose a lot. All at once. So, what do we do? How do we go about changing this? Well, what we have come to recognize is that, well, 2 billion years of evolution, prokaryote or the proto cell to the prokaryote to the beginning of us eukaryotes onto what we have now here today. Ooh, changing light there. Don't look so flushed out, I guess. Well, we have evolved mechanisms so that instead of one big transformation with one giant tax bill, thanks to that second law of thermodynamics, we now do everything in small little increments, small little changes. Yes, this doing things as small little changes means there's a lot more going on, lots of little plates to keep spinning in the air, but means the tax bill, how much you owe to the universe, thanks to that second law of thermodynamics, can and will be minimized. Everything going on in your cells, the breakdown. Well, if all goes according to evolutionary plan, the breakdown of all the things you take in, somewhere along the way, the majority of those are going to be exergonic, meaning there's excess energy, low delta G. So that reaction could have actually occurred on its own spontaneously, but through the use of enzymes, it happens faster, cleaner, more efficiently, and under a time frame our cells can utilize. In the process of this, that whatever that reaction was occurring, since it had such a low negative delta G, there's some energy to be used elsewhere. That energy is not just going to be poof, transferred one place to another. No, it's going to be taken as excited electrons. So where NAD, NADP, FAD come into play. Ultimately to make ATP. ATP is a nice short-term storage. Okay, long-term storage of energy, that's when you're going to be looking at lipids, lipid droplets and adipocytes, things like that. So everything is about conserving energy. Everything we're going to see is also about the game of hot potato. Who's got the electron? Where did it come from? Where is it going? And it's not just any electron. It's going to be an electron in a high energy or an excited state. The transferring of that electron from one enzyme, one protein to another, can allow that protein, that enzyme to bleed off a little energy to use it to do something. Don't know what. We'll see. What we have done is we've studied all the different types of reactions you see here to the center of your screen. You go and you look at this, figure 14.1 in the textbook where you can see it a little up close, a little more personally. You will see that going through and the breakdown of numerous molecules that will be found in a cell. Everything from different from different carbon-containing atoms, sulfur, methyls, different, nitrogen-containing compounds. We have gone through and we've seen the donor and the accepting reactions, how much energy in voltage you could get out of this. Up at the top, along with the delta G, the greater the negative the number, more electronegative that reaction would be, which means more energy is going to come about in those electrons that are being passed. That's why we're going to see throughout this chapter things that are using, utilizing carbon, organic molecules, as opposed to nitrogen or inorganic compounds, are able to get electrons that have a little bit of higher energy state, thus can do just a little bit more with them. But that second law is a problem. So we can't just go through and keep plucking off here, plucking off there, and attaching and moving things willy-nilly. We have to have it under a process. A couple billion years of evolution have allowed us to couple the taking of those excited electrons and using that for ATP synthesis, the making of ATP. ATP is a little more stable. Okay, well, actually, it's quite, molecularly, it is a lot more stable than what you're going to find, NADPH or FADH2 and those excited electrons. Because you think about it, NADP and FAD, their whole point of being is to take the electron and then give the electron up. ATP, no, it's a covalent bond. It's a covalent bond that forms between that second and that third phosphate, between the beta and the gamma phosphate. So it's more stable. I mean, keep in mind, everything here is relative. So it is, quote unquote, more stable. So we can keep it around. We can then utilize it in more reactions. Knowing where that game of hot potato, that I like to call it in all of my biology courses, drives people nuts, is that, that hot potato, that electron, it's all a matter of redox reactions, reductions and oxidations. Where'd the electron come from? Where is it going? In the process of taking it from one molecule to another, who got oxidized, who's being reduced, you're taking this electron from a high energy state, transferring it in a high, and then some of that energy to be utilized to do blank, something else. By going and looking at the reducing potential, we can determine, are certain molecules, organic or inorganic, going to be have a tendency to be donating, giving up excited electrons, or accepting electrons? Again, hot potato. Who's got it? Who did it come from? When we look back at that figure 1, and you see that there's different reactions on those scales. The greater the amount of energy, when we take the two halves of a reaction and we go and we see where they fall on the scale. If a reaction is two molecules that are close on that scale, yeah, there's still going to be some energy in that excited electron, but it's not going to be as great when you're looking at two halves of a reaction. A plus B yields C plus D, okay? So A and C, B and D, okay? Those are your two halves that I'm talking about. So A to C, going to be very, very low. B and D, that part, are they close? Well, that means that there's not going to be, there's going to be energy, but just not a lot. Or are they further apart on that scale? Well, that greater the difference there means there's going to be a lot more energy being transferred over. Ultimately, though, all living cells on this planet are looking for ATP synthesis, whether it's substrate level, oxidative phosphorylation, like we talk about in intracellular cell biology, using all the cytochromes and the pumping of the hydrogen, ATP synthase, all of that. And then there's also the photophosphorylation, where you're using light energy, not to excite electrons to pump, to make ATP, like we've talked about in other courses with, you know, uh, crap. Can't think of what chlorophyll does for plants, but you know it all, photosynthesis. Wow, couldn't think of the word photosynthesis. So it's not quite photosynthesis. What you're looking at is direct excitation of the proteins that are pumping the hydrogens. Instead of having chemical reactions of splitting of water and things like that, now they're using direct photonic energy to power the pumping of hydrogens that will then flow back through an ATP synthase. Now, when we're looking at many of the different types of metabolisms, we've talked about this in a previous one, chemotroph, chemoheterotrophs, chemolithotrophs, autotrophs, autolithotrophs. Are they taking it up? Are they doing it themselves? Chemo, auto. You know, are they rebuilding, taking in, the stuff they're taking in? Are they rebuilding it, repurposing it to make self, or are they fixing carbons to, carbon dioxide, carbons from carbon dioxide to make their own organic molecules? Chemotrophs the first, lithotrophic is the second. So the chemotrophs, fermentation, okay? Remember, we humans are chemoheterotrophs. So we use organic compounds that we take up and we repurpose for our own uses. And we can do versions of fermentation. What's great about fermentation is there's no external electron acceptor. Okay, oxygen, that whole reason why you have to breathe in. Remember, the reason you need to breathe in is for oxygen to be there as the final electron acceptor after oxidative phosphorylation. No oxygen, no oxidative phosphorylation. No oxidative phosphorylation, no citric acid cycle, nowhere for the pyruvate to go, and NADH doesn't get oxidized back to NAD, which means glycolysis shuts down. So, billions of years ago, cells started evolving to utilize fermentation. It's a workaround. It's a workaround that allows for glycolysis to continue at a slower, abated pace. But ATP is going to be generated, but it's through substrate level phosphorylation. No ATP synthase, no proton motive force, no lots and lots of ATPs for the cell to use. Low levels via enzymes. Respiration, which is what I just talked about, using oxygen as the final electron acceptor, means that the excited electrons that are plucked off of carbon skeletons in glycolysis as well as the citric acid cycle, those excited electrons will then be utilized by different cytochrome complexes to pump hydrogens across a membrane. For us eukaryotes, this is in the mitochondria, from the matrix to the intermembrane space. For prokaryotes, it's going to be from the cytoplasm across their cytoplasmic membrane into the space, either between the thick peptidoglycan layer for gram positives or into the periplasmic space for gram negatives. Setting up a gradient, a differential. High hydrogens, thus high positive on one side, negative-ish on the other side, okay? It's basically a biological battery. They refer to this as a proton motive force, okay? Because you're setting up an electrochemical gradient. These protons are then going to flow back across the membrane through ATP synthase. So you're using electron energy, where those cytochromes are bleeding off little bits of the energy from that excited electron, pumping hydrogens. So you're setting up a differential, that electrochemical gradient across on either side of the membrane, the flowing of the hydrogens through ATP synthase. So it's a hydrogen movement causes the spinning of ATP synthase, which is kinetic energy. The spinning of ATP synthase causes the catalytic knobs of ATP synthase to grab a diphosphate, inorganic phosphate, slap them together, so you now have a third phosphate, the gamma phosphate, and the energy is now caught in that chemical bond. Flowing of hydrogens becomes mechanical, becomes chemical. There you go. And we have not broken first law of thermodynamics. Chemolithotrophs do a version of this respiration, but they're going to use some inorganic electron donors. Okay, those are the nitrogens, the sulfurs, the seleniums, and things we saw on table 14.1. Or, sorry, here, table 14.1. So, as you see here, not everything on this planet utilizes carbon sources like we do. Okay, sulfide, sulfur, hydrogen, ammonia, nitrite. Is the energy going to be found in these bonds the same level that you're going to find in organic molecules? No, but it's more than enough. Enough excited electrons. The delta G's are going to be nicely exergonic. That's going to allow for the making of ATP. Is the making of ATP going to be at yields comparable to what you'll see in organic metabolism? Yeah. For some of them, it will be. For the majority of them, no, it won't be. Well, that's kind of vague, because when it comes right down to it, not all organic metabolizing cellular organisms have the same level of ATP synthesis, the same level of delta G's and all of their organic, that you see here in the textbook. So, just to be vague about it. We have to remember, not everything on this planet is like us. Not everything is going to be aerobic. The majority of organisms out there are going to be anaerobic. They don't want oxygen. They don't need it. They don't want to be around it. So their final electron acceptor, because yes, they're going to do their own version of respiration. It's not going to be, it's not going to be based on oxygen. So their oxidative phosphorylation, that oxidative does not mean oxygen, it means redox. Okay? Their oxidative phosphorylation is going to be utilizing something other than O2, some nitrogen or sulfur containing compounds. Some of them even use an iron. Atom of iron in some molecule will be the final electron acceptor. Okay, not everything is like us. Very few things out there comparably are like us. Okay, they outnumber us. Keep that in mind. The O2H2O that we see in us has the greatest yield, okay? Because of the electropositivity of the reaction. Remember the half reactions where they fall on that scale. Well, this is, one of the greatest ones out there. So a lot more energy can come out of this reaction. So that's why, part of the reason why bacterial cells that utilize these anaerobic respiration, utilize things other than O2 as the final electron acceptor, struggle in the making of ATP. So they've got to do more to get, levels of ATP that are then going to be utilized by their high metabolisms. Because remember, every 15 to 30 minutes, 1 becomes 2, 2 becomes 4, whatnot. So to compensate for this, they got to stay small. Part of the reason why prokaryotic cells, our key bacteria, are all generally small. Yes, there are a few that are big, big's relative, okay? Okay, a lot of this I've already talked about, so we're just going to bounce through it real quick. So it's the reducing power of many of the organic and inorganic compounds. Where's the electrons? Who's got it? Where's it coming from? Where's it going? And when it's being moved, energy is going along with it. As I mentioned before, there are numerous carriers. They're going to be taking those excited electrons and then transferring them from point A to point B, because you can't just have an excited electron moving around a cell, because it's going to interact with way too many things. So you're going to utilize NAD or NADP and FAD, giving you NADH, NADPH, or FADH2. And chemoorganotrophs. Chemoorganotrophs. They're going to be looking at the reduction of organic compounds, and only organic compounds. Chemoheterotrophs, us, we pretty much use organic, but every so often we can slip in a little something else we can break down, utilize. It's not, don't know the numbers. Top of my head, I'd say it's 99% of the time it's going to be organic in us. But every so often, certain things that come up in your diet may be broken down. What and where, don't know, don't care. Not for this course, not for this lecture. The methods that we have characterized in living cells to couple exergonic and inorganic. Well, ATP hydrolysis has been one of the best. Requires a lot of energy to get ATP there made. It's going to require a little bit of energy to cut. But once it's cut, that terminal phosphate is cut off of ATP, yielding ADP. It's extremely exergonic, so that the energy that was utilized to form ATP, the majority of it has been stored, and the majority of that will then be released to power the next. We've also seen that there are alternate mechanisms for doing things. Reverse electron transport. So up till now, I've been keep saying, the energy from an electron is bled off by a series of proteins and things, the cytochrome complexes, that as it's bleeding off the energy will then use that to move hydrogens across and the flowing of hydrogens. Well, it turns out there's other. You can reduce NAD or NADP by the use of a proton motive force. So as hydrogens are flowing back across through a membrane, diffusion, or there's some form of a transport. This is allowing for the plucking of excited electrons. Remember, in all courses of biology, when I say excited electrons, I'm talking a lot of times hydrogens. Because remember, hydrogen's nothing more than an electron with a proton baggage. NADP becomes NADPH. We also have to take into account what is referred to as electron bifurcation, endergonic reduction of low potential, is driven by high potential exergonic reactions. Okay, makes sense. You're going to utilize some high energy releasing reaction, Some of that energy is then going to be taken and then utilized in low potential mechanisms. You'll see this with flavin enzymes, the catabolic breakdown of compound whatever, molecule whatever. In the process, flavin coenzyme will give up an electron. It's part of the overall enzyme. Okay, it's a coenzyme. Remember, coenzyme is a smaller molecule than the enzyme itself that either has to associate with the enzyme or associate in the active site of the enzyme. As it's there, the substrate's being broken down, it's an exergonic reaction. Well, during that exergonic reaction, the flavin is going to release an electron. An electron that may or may not be in a very high energy state, which will then be transferred over to something else. Ferredoxin or NAD, FAD, whatever. So the breaking of something, as you see on the left, the donor. Hydrogen formate, NADP, NADPH, NADH. That electron will then go through FAD and then be passed on. Low potential, high potential. Where's it coming from? Where is it going? Now, as the energy is being stripped from proteins broken down to amino acids broken down into their constituents. Well, are those constituents or those amino acids the nucleotides, the sugars of the nucleotides, that phosphate group, the nitrogenous rings, are any of that being utilized by the cell for things other than the energy in the bonds? Are the nucleotides, the sugars, being utilized to make new nucleotides? Are the amino acids, or if not the amino acids, maybe the amino part, the nitrogen containing part of the amino acid, is that being reutilized? If they are, that is what is referred to as assimilative. Okay, things taken in, things broken down, majority of those things are then utilized to manufacture, rebuild that cell. If they're broken down and they're just energy, they're going to be released as carbon dioxide. Well, that's dissimilative. Turns out, as a side note, most of the organisms that do some form of CO2 fixation, they're assimilative in nature. They are going to take in things, they're going to reuse things. They're going to break them down, reuse them, and rebuild themselves.